understand in our lab why this F18 positive entero-toxigenic E. coli or shiga toxigenic E. coli always occur the second week, the third week after weaning, and not the first week after weaning like F4 ETEC. And I already explained that one of the reasons is the presence of the receptor, which is not there at birth, which is appearing at weaning but not optimally expressed, and then starts to become optimally expressed 4 or 5 weeks after birth. The second reason is the intimate adhesion of the fimbria with the mucosa. F18 fimbria need to interact really with the cells of the mucosa, and normally cells are not free. The cells are covered by a mucus layer. So one of the ideas, one of the hypotheses we tested in our lab was to see if animals are first infected with an entero-toxigenic E. coli strain, if that could make them more susceptible to infection with shiga toxigenic E. coli strain. And so we did some experiments, but came then to the setup that we injected the piglets in the intestine with the LT enterotoxin that is causing secretion. And we tested different dosages and chose a dose that just dilutes the mucus layer. So not really clears the mucus layer, but dilutes the mucus layer. And if we infect them 6 hours later with F18 shiga toxigenic E. coli, then you see that the animals that first receive LT enterotoxin are excreting the shiga toxigenic E. coli strain for a longer period than when they don't receive a toxin. So also, if you look at colonization in the intestine, the gray bars present the animals that have been first treated with the LT enterotoxin and then infected. And you can see that the ones that have been pre-treated with the LT enterotoxin show a much stronger colonization than when there is no LT treatment. And this is also expressed in the duration of excretion. So from 5 days after infection, the ones that have been treated with LT excrete significantly higher, so significantly higher quantities of the bacteria. This is the cartoon we made of that. So first F4 E. coli comes immediately after weaning on a farm, and then this produces toxins which dilute the mucus. The receptor for the F18 bacteria is available. The bacteria start to colonize, and you see these clusters occurring on the mucosa, and disease is appearing.